Good morning, everyone. This is John Heerdink. I am the founder and managing director of Tribe Public. Thanks so much for joining us today. As many of you know, and that have joined us in the past, we are a group that gets together across the world through webinar-based events. And now, since we're pushing forward post-COVID, if you will, we've started to do live events on the West Coast again, where we bring forward the CEOs of the companies that the tribe cares about through our wish list process at tribepublic.com. Note that I also run an investment advisor called Vista Partners. Vista Partners is an investor in Atossa. Please review our full set of disclaimers at both sites, as we also work as an advisor to Atossa and have done so over the last couple of years. And we're excited to have our speakers today for our program, which is going to be about 30 minutes. If for any reason you want to review what we speak with Dr. Quay and Kyle Gusay, the CEO and CFO of Atossa, after the event, we'll be publishing the video of this event at the Tribe Public YouTube channel for all to view as early as possible after we get the recording set. I'd like to again thank all of you for joining us today. And the event title today is Atossa Therapeutics, Redefining Breast Cancer Prevention and Treatment. Again, this will be co-hosted by the world-renowned scientist in the picture there, author, entrepreneur, Stephen Quay, MD, PhD, founder, CEO of the Seattle-based Atossa Therapeutics. The symbol again is Nasdaq, on the Nasdaq, ATOS. And Atossa is a clinical stage biopharmaceutical company developing innovative proprietary medicines to address significant unmet needs in cancer. We also have esteemed colleague Kyle Gusay, the CFO of Atossa, pictured for this event, and he'll be addressing some questions. A little bit of background about Dr. Quay. He's had an amazing career and has a named inventor on 87 U.S. patents, 130 pending U.S. patent applications, named inventor on patents covering 5 pharmaceutical products that are approved by the FDA. Dr. Quay, an MD became an MD in 1977 and a PhD in 75 from the University of Michigan. Received his BA degree in biology, chemistry, and mathematics from Western Michigan University in 1971. He was selected to serve the company's board of directors at Atossa because of his role as a founder of the company, and as well as his qualifications as physician and a principal researcher overseeing the clinical and regulatory development of the company's pharmaceutical programs. Thanks again for coming to this event, and why don't we get started straight away, and we'll start asking some questions for Dr. Quay, but maybe Dr. Quay, can you give us a little bit of background on how Atossa was started?
Sure. Well, so John, it was started in the context of looking at the amazing success that was achieved in cervical cancer when there was a test that could see the progress from normal to hyperplasia to atypical to carcinoma in situ. And that PAP test, that was called, took the incidence of cervical cancer from 110,000 a year down to about 15,000 a year. We now know that a virus is involved. There's a vaccination for it. So we're basically looking at the elimination of cervical cancer. So about over a decade ago, I said, why can't we do the same thing with breast cancer? And the key process, the two-step process is, what do you measure before you have cancer in the breast? And can you modulate the changes that are going on, the biology that's going on, before it crosses the line and becomes cancerous? And so the answer to that is yes and yes. The first issue is that by looking at the density of the breast, that is the background parenchyma. So when you do a mammogram, you're really looking for grains of salt, little salt grains, which represent dying cancer. So that's what the mammogram was developed for. Is there cancer or is there not cancer? And then after about 20 years, someone looked at it and said, hey, if I look at just the background, is the background mostly black or is the background mostly white? These are all women with no cancer. If I follow them for 10 years, guess what? The women that are all white tend to have about 8 to 10 times more cancers. So that's exactly the analogy that was done with the cervical pap smear. So with that backdrop, we're now saying, well, can we modulate those women with high density? Can we make the density lower? And for lots of reasons, we think that is a biologically tunable property. And so the clinical trial we're doing in Sweden at the Karolinska Institute, the place where the Nobel Prize is awarded, is looking at exactly that. A woman with high density, if she takes endoxifen or placebo for 6 months, will she see a change with endoxifen? Not with placebo. And then if you wait 18 months off of drug to see if it's durable, does the density reduction stay down? So it's probably, as you say, I've invented 5 drugs. They've been used in 80 million people, but this program to prevent prevent upwards of 200,000 breast cancers a year would be the capstone of all the things I've done in my career.
That's fantastic. Thanks for your continued research in this area. And as I've always been very excited about the program, it's hit home, very close to home. As you know, when we originally met, my mom was stricken with the situation and breast cancer, and is surviving and still is today. But it's this unaddressed need here is there's still work to be done, and I'm so happy to hear that you're going to continue on this work, this path. So the company, as I understand it, has transitioned from early detection to prevention and treatment. Can you give us an overview of Z-Endoxifin that your proprietary Z-Endoxifin?
Sure. So once you know that you can measure density, and once you know that the density is under control of the estrogen that a woman produces in her ovaries, then what you want to do is find something that blocks the estrogen. Tamoxifen is the classic example of that, and was really the first hormone modulation therapy that was brought into the cancer field. Tamoxifen, however, in the body makes about 21 different chemicals. It's really quite a pleomorphic drug, if you will. And so by teasing out which of the 21 had the greatest activity, we and others were able to show that Z-Endoxifin was the real agent out of all the 21 that was doing, well, we quantified it, probably 95% of the effect. So we now have patents on both the chemical synthesis of Z-Endoxifin and then the way you have to formulate it to get it from an oral tablet into the bloodstream, because it's acid sensitive. It gets destroyed by the stomach acid. So we have 2 complementary patents on the white powder, as it were, and on how you would tabulate it, to allow us to really push the frontiers of what endoxifen can do. The buzzword in biotech is having a pipeline in a product, and some people have to kind of stretch it. But I think if you see our programs from prevention of breast cancer to neoadjuvant treatment to adjuvant treatment, we really are bookending breast cancer. And by the way, there are some other conditions in the body that are estrogen driven that we may explore.
That's exciting. It's truly a platform to really build an amazing company off of, and it's exciting that you continue to find ways to carve out a significant IP portfolio as well around endoxifen. If you could continue with, as you mentioned, as you're investigating Z-Endoxifin in 2 settings, neoadjuvant and mammographic density, breast density, which is is more of a breast cancer prevention play? Can you explain both again?
Sorry, I didn't hear that. You broke up technically.
Yeah, I'm sorry. It says, basically what I'm looking for is, can you explain in the 2 settings, neoadjuvant treatment and mammographic breast density, which is more of a breast cancer prevention play? Or can you explain both?
Yeah, well, no. So treatment of density, because high density is a condition that is not cancer, but women that have it have a very high incidence of cancer, it would be considered a prevention process. Neoadjuvant is by definition a treatment that's given to women from the time of diagnosis to the time of definitive therapy, which still currently is surgery and plus radiation if it's needed for local disease. So that window can be as short as about 3 weeks or as long as 6 months in some settings. And it's kind of ironic that the 2/3 to 3/4 of breast cancers that are the most common, that is the ER-positive cancers, are really not treated in the neoadjuvant setting. We kind of skip that period because every time someone has tried a drug there, it hasn't worked. My hypothesis was that in the case of tamoxifen, for example, it takes so long to get therapeutic levels of the tiny amount of endoxifin that's being produced from tamoxifen. 6 weeks is really the steady state levels, that you've moved on to the surgery and radiation by the time you get there. With our Z-Endoxifin, within 4 hours, you have blood levels, and within 24 hours, you have therapeutic blood levels. So basically, a woman can go from a needle biopsy of a lesion. The bad news is it's cancer. The good news is here's a prescription. Go to your pharmacist and start taking endoxifen, because we think, based on what we hope Atossa's clinical trials will show, we think that this will slow down the tumor, maybe shrink it before we even get to surgery. So that's a really exciting window. It's an unmet medical need, again, because there is no other drug that really works in that space. And so that's kind of our focus. Now, there's a corollary focus, which is that breast cancer can occur in two women in 2 stages of their life, premenopausal, postmenopausal. So in the postmenopausal case, the ovaries have stopped making estrogen. There's a little bit of estrogen made in the breast itself. And so you don't need to do much other than block that local activity to get the cancers to really respond. Premenopausal woman is a more complicated situation because you have the ovaries willing to make as much estrogen as they need to. So if you go in with a modest suppressor of estrogen, what happens is the ovaries overpower that, and you get ovarian activity. So there's a protocol called ovarian suppression, which is common in premenopausal women in the neoadjuvant setting, and it's pretty harsh in terms of quality of life issues. The women really don't like it. Sometimes they will actually risk getting cancer rather than stay, in compliance in that particular protocol. So the other setting we're trying endoxifen is, because it is such a strong suppressor of the estrogen activity itself, we believe that it'll work in a premenopausal woman without ovarian suppression, which would be a godsend for these women in terms of their quality of life.
Yeah, that would be fantastic. Could you also speak to the sort of, if you quote unquote, market size or the patient population size in the US in both of these settings?
Yeah, well, sure. So they're big numbers. So in terms of breast density reduction, we start with a number that there's 39 to 40 million mammograms each year done in women. And that's about 80% of what we think should be the right number. So women are much better than men, frankly, and that's a whole separate conversation on their own health. And so we're getting about 80% of women who should be getting mammogram are getting it. And that's because there's a lot of financial support and programs and education around getting your annual mammogram. So 40 million is your total number there. And upwards of 10 million have high density. And so when you combine that with maybe a couple of the risk factors, you get millions of women who would be candidates for a 6-month treatment to reduce their density, which then we believe will have downturn effects on incidence of cancer. In the cancer setting itself, about 250,000 women in the US are getting cancer each year. 2/3 to 3/4 of those are estrogen receptor positive. So that's 200,000 is a good number to imagine is the treatment. So those women are going to go through neoadjuvant, they're going to have surgery, they're going to have radiation, and then they're going to have 5 years of follow-up adjuvant therapy. So the ER positive population is roughly 200,000 plus 5 years of 200,000, or about 1 million women in the US.
Great. I guess carrying on with this discussion, could you also, it's my understanding that you're currently investigating in both settings and ongoing phase 2 trials. Can you tell us about the studies that you're currently conducting? And please carry on.
Yeah, sure. Well, I mean, the study in Sweden is a placebo-controlled trial. It involves 240 women in groups divided up in that. The treatment is 6 months, and they either get a sugar pill or endoxifen. Nobody knows what they get. One of the extra benefits of endoxifen, which I will take no credit for because I didn't predict it, I predicted the opposite, is the side effect profile. So one of the issues with tamoxifen in the prevention setting, it is approved for prevention, it's used in 2% of women, is because it has a lot of side effects. And so in a population that doesn't have breast cancer, you're trying to reduce their incidence in the future, your tolerance for sort of quality of life or side effects is not very high. And so one of the things that we thought, we would have an issue with endoxifin was a similar pattern of side effects. And, knock on wood, we don't see the same incidence of what are called vasomotor symptoms, which are night sweats, flushing, overheating, basically. We don't see those with endoxifin at nearly the level you do with tamoxifen, and it's a remarkable difference. So again, at this point in time, early days in our clinical trials, but that's a really beneficial process. The process is a baseline mammogram at 6 months, a mammogram at 24 months. And the three of those are used to look at the trajectory in these women of whether there's a dramatic drop between the first and the 6 month one, and whether a drop, if it occurs, is maintained out for 24 months. There really is, we do have some preliminary data in like 20 or 30 women where if you do see a reduction with tamoxifen or other drugs like that, it seems to be pretty durable in about 85% of women. So we're expecting that to be successful, but science is science, and that's why you have to do it before you can opine on it completely. But looking at the durability will be an important factor. From a business point of view, a lot of companies want to have a drug that they give a woman for their entire life or give a patient for their entire life. And that's a business model, money-making business model. We think here the best care may be a 6 month process. And that's exciting from a physician's point of view, because a woman shouldn't think about her breast health unless she has family history until she's 40. She gets her first mammogram. If she has high density, not great, not the best day of her life. But with a 6 month prescription, if it goes down and if it stays down, we can really change their whole future. And perhaps, give them a 20-year window of reduced risk on breast cancer. Just, I work 20 or 18 out of the hours in the day, and these concepts and this opportunity is what gets me out of bed in the morning.
Thank you. Could you give us a little bit more of the specifics on how you've structured your trials here, Evangeline, and also the MBD charisma endoxifin studies?
Yeah, well, I'm not sure quite what you're saying, but all placebo-controlled trials have a randomization process that dictates which particular pathway the patients go down, and then you're kind of loath to ever break the blind, and so we've talked about it with one particular patient. Had some interesting things that we wanted to. She probably needed to go off the trial, but we didn't break the blind in that case, and you usually don't break the blind. In the Evangeline trial that Matthew Getz is conducting in other centers, it again is a randomized process to endoxifin or to ovarian suppression, which is standard of care right now. There is a what we call a PK run-in. So we know what blood levels are most effective for various things with respect to endoxifin, because there's so much experience around them. So so what we're doing is a PK run-in, where we get three to 4 weeks of drug in the women, and then we measure their blood levels. And if they're too low, we have a protocol where we go up a dose. Too high, we go down a dose. And this process is we're making progress in this, and this will really define the next stage, where we'll just do one dose, placebo versus active, for about 6 months. And the hope there is to not only suppress ovarian so that the estrogen is reduced, but perhaps get downstaging of the cancers. So the ultimate home run with the bases loaded in neoadjuvant is to take a clinical stage cancer and lower its clinical staging, which usually means it gets smaller. It makes the surgery more compact, maybe more aesthetically, more plastic surgery like, and so that's sort of again the ultimate outcome. So that's the design, John.
Yeah. Okay. Thank you. On the Evangeline trial, I believe you had your first patient enrolled in February of this year. Is that correct? And I think you're trying to pull from, is it 25 sites in the U.S.?
Yeah. And we continue to add sites. It's a, clinical trials are really complicated. So having a site say, yeah, we'd love to enter your trial, and then doing their first patient, there's 3 feet of paperwork or 2 feet of paperwork and meetings and all sorts of things in the team. So we're making good progress on that. We'll give you updates when we feel it's appropriate in the terms of the recruitment process. So we're feeling comfortable with where we're at in that trial.
Well, it would seem like I guess again, the treatment on the Evangeline is that you treat for up to 6 months prior to surgery. Is that correct?
That's correct.
That's great. So you looking forward to seeing that progress and all of this revealed as you move forward this year and beyond. So we're excited to hear about this. In the essence of time, I know we should probably continue to move along our path here. Can you speak? One of the things that's important to many investors is that you have solid patent protection. Can you speak to that?
Sure. Yeah. Want to just correct you. I have 88, not 87 patents, but who's counting? So patents are very important for pharmaceuticals, and they're really, I won't get too philosophical, but one of the only ways that a tiny company like this could ever, expect to compete with the Pfizers of the world is this patent system. And it allows us to get value for our shareholders from our inventions, because the government stands in the way of people simply, being so big they can just come in and take our stuff, so to speak. So I know there may be some, popular movements around patents and that sort of thing, but I think people should really realize that the foundation of America's technological success over 250 years is that we were the first company with a formalized patent system. It's in the Constitution, actually, of the country. So in that context, you want to have patents in kind of 3 areas in pharmaceuticals. You want to have the composition, the chemical itself. So just 5 million chemicals in the world, and endoxifin is one of them, and we own the endoxifin patent itself. You have to have methods of making that particular drug. So those are a little more complicated because you have to prove infringement going into the factory, but we have those kind of patents. Once the drug is made, it sometimes can be formulated in a patentable way, and that turned out to be the case for endoxifin. So we had to make it enterically stable so that it didn't convert from Z-Endoxifin to a mixture of Z and E, which it wants to do in acid. And the E form is completely inert. So you get, you lose 50% of your drug in 40 minutes if you don't use the patented process. And the FDA is not going to allow you, to ever not use our patented process there. And in many cases, then the final stage is that you can get methods of treatment where basically it's the doctor who's doing the infringement. So a method of treating breast cancer, the steps consisting of identifying it as ER positive, and then treating them with endoxifin. So that's been our strategy. We are, I think, among the best in patent strategies. I've had 5 litigations, all won all 5 of them, multi-million-dollar settlements for people that thought they could just steal my stuff. And we had to show them that in the court system that was not the case.
Exciting. Well, switching gears a little bit, which is also important to investors, is to understand how you're funded. Can you speak to how the company is capitalized to date and your current burn?
I could speak to it, but Kyle Goosey can do a better job, so I'll let him.
Fair enough.
Thanks, Dave. Thanks, John. Atossa is, I'll say, very well capitalized for a small biotech company. Around COVID time, we raised about $160 million in the capital markets when the capital markets were very, very active. And as a result, at the end of 2022, we had over $100 million in the bank. To put things in perspective, that's multiple years of cash for Atossa. So you'll notice our auditors do not have a qualification for going concern for Atossa, which is very common in small biotech companies. So that's our funding situation right now.
Gotcha. And can you speak to the historic burn?
Yeah, John. We historically have burned $1 to $2 million in cash per month. That'll go up a little bit as we continue to enroll in these phase 2 clinical studies that Dr. Quay was talking about. But that's sort of our historic burn rate. So we feel very, very confident about our cash position.
Okay. Yeah. Well, with that significant cash position, what are you also continuing to look at any potential acquisitions or investments?
The short answer is yes. We're opportunistic. We have enough cash that we could invest it in something that looks sufficiently compelling. We did make an investment of $4.7 million in a venture capital backed CAR T company here in the United States, acquired a 19% interest in them. And so we're constantly evaluating other opportunities. The challenge is finding something that's as exciting as the things that we're working on.
Mm-hmm.
We don't want to dilute our pipeline and our programs by acquiring something that's not as compelling as what we have. So we've set the benchmark pretty high, but we are opportunistic.
Yeah, and I guess another way I'd say that, and Kyle knows this because we work together so tightly, John, is the investor should imagine that we probably on any given day have one potential acquisition or program that we're looking at. And that's kind of been consistent since probably, maybe July or June of last year. So something comes up, we do some due diligence on it. We spend various amounts of times on any one given program, but it kind of has been historically, we see something, we don't really like it, we say, okay. And then like the next week, someone, because we have people, and we find things on our own, but we have a lot of people sending us incomings, we start the process again. So we are always active in that space, but just repeating what Kyle said, endoxifen is one of the greatest opportunities in breast cancer for patients. And we just want to get that thing across the goal line.
With a follow-on, we've got several additional questions from the tribe as given us access to you today. Building on one of the questions that came in says the Australia Z-Endoxifin trial was halted early due to tremendous success, which is likely what garnered the attention of many current shareholders. As we eagerly await updates from Evangeline, if similar success is observed in early participants, could the trial be halted as well? Or can you speak to that?
Yeah. So the basis for that is when you perform the statistical design part of a clinical trial and set the number of patients, you built into that an expected outcome, which is based on your previous information. Mm-hmm. So when we did the trial in August in Australia, we used what we expected to be the experience from other drugs in that, and endoxifen blew it out of the park. So what that means is that you can stop the trial early in Australia because you have all the efficacy you're ever going to see. Now, you come to the Evangeline trial, well, guess what we have to do by kind of convention is we have to take that efficacy from Australia and bring it into the statistical evaluation of the number of patients in Evangeline to get the results. So the number of patients is baked in to that very high level of success. So I don't expect to be able to stop the trial early because of that particular finding. The bar effectively gets raised in the trial itself. But what it does mean is our data will be extremely robust should we have the similar results.
Okay, great. Follow on to Z-Endoxifin says, another question is, research into the history of Z-Endoxifin shows that previous trials have taken place investigating its effects, not just on breast cancer, but desmoid and gynecologic tumors as well. One P1 trial concluded in 2020 was funded by the National Cancer Institute and showed encouraging results. Has Atossa considered seeking funding from the NCI for its Z-Endoxifin studies and/or expanding its trials to include other forms of cancer?
Yeah, so I think the short answer is that yes. The National Cancer Institute is constantly funding both in prevention and in treatment for cancer trials. And as all companies in the oncology space do, are looking for opportunities to let them assist in the funding, because it's non-dilutive funding. It's not free money, but it's very attractive. As for other cancers, so again, if you think about it, when you give estrogen to a cell, 15,000 of the 20,000 transcripts, enzyme activities going on in the cell are changed within 15 minutes. So estrogen is perhaps one of the most pleomorphic effectors of cells that we know. So we are looking at estrogen-driven mechanisms for other diseases for which blocking that activity could have a salutary effect like it does in breast cancer. And that is probably that investigation or that thinking is a high priority for us this year.
Okay. Can you also provide us with an enrollment update on the 2 neoadjuvant trials and an idea of when we expect to see data from each?
Yeah, it's kind of the same question, John, so you're not going to trick me. We can't give you updates on the neoadjuvant trial. We said when they start, and we will give you an update at an appropriate point in time, which is typically a data read. So, I mean, it's great to know if you've done 20 patients or 15 patients or whatever it is, but it's great to know. But it's great to know, hey, we're doing a look at the data for the following reasons. Here's the results, and what's the next step. So, as soon as we can, we'll be giving you that information. We understand your interest because it's only a millisecond behind our interest.
Got it. And then another question is, what is the best case scenario for the neoadjuvant setting? Is the goal to halt tumor growth or shrink the tumor, or is it more about reducing the risk of reoccurrence after surgery?
Yeah, no, that's a great question, and it's really quite specific, and I think I gave the answer earlier, which is the home run with the bases loaded for neoadjuvant treatment is a clinical downstaging of the cancer. So the clinicians have numbers and letters associated with the clinical stage of the disease, and so sometimes the radiologist can say, well, the tumor got smaller, and that's really good, but it's the clinician who says, yeah, but you didn't say it didn't change the stage of it. So best case scenario is to downstage cancers. And then that kind of puts them on slightly different pathways for what their ultimate treatment would be. So that is every neoadjuvant company's goal for the highest level.
Okay, thank you. And then lastly, will Atossa pursue the 505(b)(2) pathway for Z-Endoxifin approval at any point, or is that in the realm?
Yeah, so to understand what that means, just there are three flavors of FDA approvals: 505(b)(1), (b)(2), and J. J is generics. (b)(1) is a brand new drug that the FDA has never seen before, and the company owns all of the safety and all of the efficacy data by their ownership. They submit that and they get approval. 505(b)(2) is a very interesting hybrid where we will own our data, but we're asking the FDA to reference data they may have that we don't even know about with respect to endoxifen, because it's a metabolite of tamoxifen. So one of the categories for 505(b)(2) allowance is to be a metabolite of an already approved drug. We check that box. And the advantage is we do get credit for the work that's already been done on endoxifen and or tamoxifen that we don't know about that's inside the FDA. And there's some market exclusivity issues. We have patents, so that's less important for us, but it still is a nice, have if we were to, sell the endoxifen program to a big pharmaceutical company. I'd make a lot of that, of the value of that market exclusivity and the overall transaction, those kind of things. So, yeah, we would definitely pursue 505(b)(2).
Okay, thank you. This is probably directed more towards Kyle. The question is, what was the rationale for change in bylaws to lower the quorum requirement to 33%? And how does this impact shareholder voting moving forward?
Yeah, sure, John. So historically, to achieve a quorum at our stockholder meetings, we needed a majority of the shares represented in voting. We recently reduced that to a lower amount, 1/3 of the shares. And the purpose is simply to make it easier to establish a quorum so that we can have stockholder meetings. And so it's to facilitate meetings of our stockholders. It's a effective now is effective for today's meeting, and it will be effective going forward. So it's simply to facilitate establishing a quorum for our stockholder meetings.
Okay.
Yeah, I mean, I think, John, it's actually a response to some of the changes in the way brokerage firms decide how to vote shares and those sorts of things. So, you know, many companies have some of these challenges because in the last, say, 5 years, there have been differences in what shares get voted by the brokers versus the owners, and whether if you loan your shares, you don't vote them, and those kind of real technical things.
I see. Thank you. The stock currently is under $1. Is there a possibility we could expect a reverse split at any point in order to comply with Nasdaq?
Yeah, I'll address that one, John. I mean, our plan to address the stock price is to drive stockholder value with the programs that we're talking about, and to drive stock price movement with strong results on the things we're working on. So that's really what we're focused on. The stock, if it continues to trade under $1 and we get close to the Nasdaq deadline, we'll evaluate doing a reverse stock split then. But we'd prefer not to. We'd prefer to drive out of this dollar stock threshold through fundamental work here at the company.
Yeah, thank you. Well, gentlemen, we're right at the edge. Probably need to be cutting us off, but I want to give you one more chance to sort of sum up, Dr. Quay, anything that you would like to leave with us at this point before we sign off today.
Yeah, so I mean, I think that we've been working on Endoxifin for a number of years. Our patents are starting to be issued. You may see more patents in the area, which not just further strengthens our position there. Our activities, both at the Mayo Clinic and at the Karolinska Institute in Sweden, are beginning to generate, how can I say it, a buzz in the oncology community. So one of the very gratifying things for me is to begin to think about working with some of the true, top of the pyramid KOLs with respect to developing this drug, seeing its increased potential within treatment pathways in breast cancer is truly exciting, and really, justifies a portfolio and a product proposal. And again, I'm a science guy probably at the end of the day. So seeing new opportunities for other estrogen-related processes that endoxifin may have a role in has been an important part of my time late last year, this year, more into the year of the future. So I'll just reiterate, I mean, I really don't want to reverse the stock. We think we have so much value in what we have that about, and what we have that will be rolling out as in a staggered process, an appropriate process over the next year or two. That I think the true value that when people can see what we have will be expressed in a stock price that's much higher than 60 cents a share.
Thank you. It's exciting to be, in front of you two today at this point. A company that has significant cash in this market, as we all know, is in a runway for several years. With that, is in a certain power position to look at acquisitions, as we spoke about, to look at developing their respective programs, an exciting platform with very significant markets that are unmet. It's truly exciting to be in front of you today. Again, reminders here, folks. This again, Atossa trades on the Nasdaq, symbol ATOS. We want to thank Dr. Quay and Kyle Gucci, the CFO of Atossa, to date, for sharing their insights and addressing many of our questions. And note that the video of this event will be up soon this week at the Tribe Public YouTube channel if you'd like to review any of the comments that they've made and in the presentation today. We want to thank you. And if you have any questions, you can also fire those back at me. And if you want to see Dr. Quay and Kyle back on Tribe at some point, please submit your interest through the wish list process at the Tribe Public YouTube channel. I'm sorry, website, which is www.tribe.com. tribe, TRIBE public.com, and look forward to hearing from you. Thanks for being the best part of the tribe. And thank you again, Dr. Quay and Kyle, for joining us today. Have a great rest of the week.
Thanks, John. Thanks, everybody.